How's it going, everyone? My name is Akash, and in today's video we're going to be learning all about acetylcholine. So acetylcholine is a messenger molecule, and it's polar, and it's what we call a neurotransmitter. That means that this messenger molecule is produced and synthesized and stored inside neurons, and then it's released from neurons and goes off to other cells to produce responses. Now, in this video, we're going to be focusing on the cell signaling pathway of acetylcholine. Now, in our first ever episode, we established that this is our universal cell signaling pathway framework. So we're going to first explore how acetylcholine is produced, then how acetylcholine is packaged, then how it gets released from its storage or packaging and goes off to produce a response, and then we're going to look at how acetylcholine's responses are terminated to prevent them from getting out of control. Let's get into it. Alright, so before we get ahead of ourselves, I do want to mention that I think this video correlates really heavily with some of our past ones, and I think it would be beneficial to maybe check those out before diving into this one. So as I said, our first ever video focuses on this cell signaling pathway. I think that's definitely one that you should check out first. And also in the last three videos, we've looked at general mechanisms of packaging of messenger molecules, and how they get released, and then how responses get terminated. And this would also really help just to kind of understand as we go through the cell signaling pathway specifically to acetylcholine. So I will link these 4 videos down below in the description, and I'll have time cards up at the top as I mention each one throughout the video. But yeah, so if you've watched all those and you're feeling ready, let's get into it. Okay, so the first step in the cell signaling framework for acetylcholine is production. So inside your nerve cell or neuron here, acetylcholine needs to get produced somehow. And the enzyme that produces acetylcholine is called choline acetyltransferase. And this can be shortened to the acronym CHAT. So choline acetyltransferase. And this enzyme is present inside your neuron. So let's write CHAT here, right? So what CHAT does, choline acetyltransferase, is it combines 2 different things to produce your desired messenger molecule of acetylcholine. The first thing that it uses is something called choline. And the second component is called acetyl-CoA. Now, acetyl-CoA is produced in your mitochondria. Now, mitochondria, we all know it's the powerhouse of the cell, and it's found in different cells. So, let's say in your neuron cell here, you have a mitochondria, right? That can produce acetyl-CoA. So, what choline acetyltransferase does is it combines these 2 parts together. It combines them and adds them together to form acetylcholine. So, it combines choline and acetyl-CoA to form acetylcholine. And if you look at the names, it makes sense because choline acetyltransferase, as the name suggests, it transfers an acetyl group to your choline. And by transferring an acetyl to your choline, it creates acetylcholine. And you can see that the acetyl parts from acetylcholine comes from acetyl-CoA, whereas your choline parts comes from here, your choline. So it just combines them together to get your messenger molecule. Very simple. So now we've completed our first step of production. But now that we've produced acetylcholine, we need to somehow store it and have it packaged inside your neuron. We don't want it to just let it out right away. We want to keep it nice and safe until it can be released when it's really needed. So let's take a look at our next step of packaging. So in order to package acetylcholine or other neurotransmitters, we use this storage compartment, shown by this yellow circle, and this is called a vesicle. So we somehow need to get acetylcholine into this vesicle so that it can be stored and kept for later on. And in order to help with this process, we have 2 enzymes as part of this vesicle to help us out. Now, we already took a look at the system in our video dedicated entirely to packaging mechanisms, so I'd really recommend watching that one, but we'll briefly go over it again. Your ATPase enzyme, what it does is it pumps protons from outside your vesicle into the inside. So now you have lots of protons inside your vesicle and lots of positive charge. And this creates what's called the proton motive force, because basically this accumulation of protons and positive charge inside the vesicle can be used as a driving force to pump acetylcholine inside the cell. And that part is done by our second enzyme, VAT. So what VAT does is, now that you have all these protons inside the cell and it's very positive, it can exchange these positive protons for your cationic, positively charged acetylcholine. So it can exchange this positive thing for this positive thing. So it can pump out your proton and pump in acetylcholine. And in doing so, it can basically replace these protons with acetylcholine. And now, look at that. You have acetylcholine inside your vesicle, nice, packaged, and stored away for later use. So that covers the second step of our signaling pathway of packaging. Now, before we go on to the next step of release, this is something that we've mentioned again in past videos, but I'll reiterate it again. The whole reason that we're able to package acetylcholine is because this messenger molecule is polar, and polar things are lipophobic, meaning they don't like fats. And what your membrane is made of lipids, so your acetylcholine is not going to want to go near this membrane. It's going to want to stay far away from this lipid membrane because acetylcholine doesn't like lipids. So it's going to stay in here, nice and happy inside the vesicle without trying to pass through the membrane. And that because of that, it's able to be stored. And being able to be stored is useful because you can store a large amount of it, and then when you really need it, you can release it all at once to go off and produce a big response. So that covers packaging. Let's take a look at release. Okay, so as I said before, we already covered release mechanisms in depth in a past video, and I'd really recommend watching that before getting into this one, but we'll go over again release mechanism of acetylcholine very briefly. So here you have your acetylcholine inside your vesicle all packaged up now, but as you can see, attached to the membrane of your vesicle, you have different proteins, and these are called VAMP or V-snare proteins. You can remember they both start with the letter V, which is associated with vesicle. Whereas the proteins over here, these are called SNAP slash T-snare proteins. So two sets of different proteins, one on your vesicle and one on the cell membrane of your neuron. Now, as you can see here, I've written out the 4 different steps of release. So the first one, translocation, is simple. Your vesicle just has to move over to closer to your cell membrane. That's translocation. It just moves over closer to the cell membrane, and then when it does, your VAMP and SNAP proteins, so then after it moves closer, your VAMP proteins and your SNAP proteins can link up together. They can attach. And that is called tethering. Now, once your VAMP and SNAP proteins are tethered together, then comes docking and priming. Your VAMP and SNAP proteins can continue to interact, and they can interact together and get ready for the final release step. So this usually involves extra interactions between your VAMP and SNAP proteins. And by the end of docking and priming, I'm just going to redraw this real quick. By the end of it, your vesicle is likely much closer to your cell membrane, and as you can see, your SNAP proteins and your VAMP proteins are heavily interacting with each other. So now it's almost ready for the final step of fusion/exocytosis. So what happens is your vesicle, which contains acetylcholine, the membrane of your vesicle fuses together with the membrane of your neuron cell. And when these membranes fuse together, the contents of your vesicle essentially spill out outside of the neuron, so they get released outwards. So your two membranes, they fuse together. So it looks something like this. Form something like that. And then your acetylcholine can escape. And that's the last step, fusion/exocytosis, because when the two membranes fuse together, then the the messenger molecules inside, in this case acetylcholine, are exocytosed, and they can leave the vesicle and leave the cell and go out into the synapse. Because remember, this is a neuron, right? And neurons they communicate with each other via a gap called the synapse or synaptic cleft. So now your messenger molecules have been released into the synaptic cleft, and then they can go off to other neurons, or maybe they can go off to other types of cells like muscles or glands or whatever and produce responses. So that covers the release step. So remember again, 4 steps. Translocation is where your vesicle moves closer to your cell membrane. Tethering is where your VAMP and SNAP proteins start to link up. Docking and priming is where the proteins continue to interact and get ready for the final step of the fusion of your membranes and exocytosis of your messenger molecule. Again, I know this step was a little bit more rushed, but if you want to look at it in depth, be sure to check out my past video dedicated all to release mechanisms. I think it will make it a lot more clear. But that basically covers the release of acetylcholine. Now let's take a look at, now that acetylcholine is released, it can produce a response. Okay, so now that your acetylcholine has been released from your neuron and it's out into the synapse, it can go and bind onto different types of receptors to do different types of responses. And your first type of receptor is called muscarinic receptors. And muscarinic receptors, basically these are GPCRs. Now, in the past, we made a video entirely dedicated to GPCRs, and we also made videos dedicated to different types of G proteins like GS, GI, GQ proteins. I'd really recommend watching all of these as well, but little recap. So muscarinic receptors, right? Let's do a little box to show that we're only going to focus on muscarinic receptors for now. So basically, muscarinic receptors, we know that they're GPCRs, and we know GPCRs are receptors bound in the cell membrane. They have an extracellular and an intracellular portion, and it goes back and forth. And so acetylcholine can come and bind to these muscarinic GPCRs receptors. And when it does, it activates G proteins inside the cell. And in fact, it can activate 2 different types of G proteins depending on your muscarinic receptor. So I'm going to show a second cell with another GPCR and acetylcholine coming and binding to this. So essentially, you have 5 types of muscarinic receptors, and these can be denoted as M1, M2, like muscarinic 1, muscarinic 2, muscarinic 3, M4, and M5. So the odd number ones, M1, 3, and 5, these muscarinic receptors all activate the same G protein when acetylcholine comes and binds onto them. And the name of this G protein is the GQ protein. We've made a video dedicated all to GQ proteins. Be sure to check that one out. And the even number of muscarinic receptors, so M2 and M4, these ones, when acetylcholine comes and binds to these muscarinic receptors, instead of activating GQ proteins, you instead activate GI proteins. So 2 different types of G proteins. And then when they're activated, GQ proteins can go off and activate an enzyme called phospholipase C, which produces your second messengers TAG plus IP3. And that can do responses. On the other hand, your GI protein, it lowers activity of your enzyme adenyl cyclase. So I'll put a minus there because it lowers activity, and then that lowers production of your second messenger, CAMP. So while muscarinic receptors 1, 3, and 5 activate GQ proteins, which activate an enzyme phospholipase C to produce these 2 second messengers, on the other hand, your muscarinic 2 and muscarinic 4 receptors, when they get activated by acetylcholine, you instead activate G proteins, which lowers adenyl cyclase activity and lowers production of your CAMP second messenger. Again, I know we're going fast, I know this may seem complicated, but be sure to check out my last videos dedicated entirely to GQ and GI proteins. So, muscarinic receptors, which are your GPCR receptors, they can be found through a variety of tissues throughout the body. They can be found in cardiac muscle, they can be found in smooth muscle. They can be found in different glands, such as sweat glands, and vascular endothelium, which basically means the endothelial layer of blood vessels. So they can be found in all of these different types of tissues, plus many, many more. And they can do a variety of responses, and one very common response is the contraction of muscles. So for example, acetylcholine can cause contraction of your airway muscles, which can make it more difficult to breathe. Now, we're going to go really in depth with acetylcholine responses in a future video where we look at your sympathetic and parasympathetic nervous system, because acetylcholine works with a few other messenger molecules like norepinephrine and dopamine to regulate your sympathetic and parasympathetic systems. It's very involved in these systems. So we're first going to learn about these each of these individual messenger molecules. Then we're going to see how they interact to control your sympathetic parasympathetic system. But for now, just know that these are your muscarinic receptors, and they're found in these tissues plus many more, and can do various responses such as causing muscle contraction. So that was muscarinic receptors. Now let's focus on the second type of receptor that acetylcholine can bind to, which are your nicotinic receptors. Okay, so nicotinic receptors, unlike muscarinic receptors, these aren't GPCRs, but they, when these receptors are activated by acetylcholine, they open up ion channels. So if you have a cell and you have a nicotinic receptor, and your acetylcholine comes and binds to it, then it can open up ion channels to allow different ions to enter. And these can be positive ions or negative ions, and this can have different effects inside the cell. So in fact, there are two main types. Let me just box this off because we're going to focus on nicotinic receptors. There's two main types of nicotinic receptors. There's NMJ, which stands for neuromuscular junction, and then you have neuronal nicotinic receptors. So first, let's focus on neuromuscular junction receptors. So these types of nicotinic receptors, as the name suggests, are found at the junctions between neurons and muscles. So you have a neuron, which releases acetylcholine, and this can go and act on muscles. So it goes and binds to these receptors, and in fact, with the NMJ receptors, you need two acetylcholines to go and bind to these receptors. And when two acetylcholines are gone and bound to it, then it can open up ion channels to allow cations to enter, cations such as potassium ions or sodium ions, but positively charged ions, which increases the charge, which makes the inside of the muscle cell more positive, and this causes depolarization and can cause contraction of the muscle. So these neuromuscular junction receptors can cause muscle contraction, and this is more so for skeletal muscles. Skeletal muscles are voluntary muscles such as, your fingers, your hands, your feet, moving all of those from your own free will voluntarily. This is done through acetylcholine binding to nicotinic NMJ receptors and depolarizing these muscle fibers, causing contraction. So that's neuromuscular junctions, and now let's look at your neuronal nicotinic receptors. So unlike NMJ receptors, neuronal nicotinic receptors are found on neurons and also something else called ganglia, which is the plural for ganglion. So ganglions are basically, you can think of them as relay stations between neurons. So you have preganglionic neurons, and then you have your ganglion in the middle, and then you have postganglionic neurons. So it's like a relay station for communication between them. So I know this isn't the best explanation. We don't really know what ganglions are, what they do. But as I mentioned before, acetylcholine is very much involved in the autonomic nervous system and, parasympathetic and sympathetic nervous systems. And the neuronal nicotinic receptors and ganglia are very, very much involved in this parasympathetic and sympathetic systems. So in the future video where we go in depth with these systems and we learn more about them, it's going to become a lot more clear about what these nicotinic neuronal receptors do. But for now, think of neuromuscular junction nicotinic receptors as being the type that activate muscles and cause muscle contraction, and then neuronal nicotinic receptors as being involved with neurons and ganglia, which are basically relay stations between neurons. So that basically covers response. We've looked at how acetylcholine can bind onto two different types of receptors. The first, muscarinic receptors, which are GPCRs, bound to a cell membrane. And you have 5 different types, which can activate 2 different G proteins. So just to recap, GQ proteins and GI proteins, the first of which can be activated by your muscarinic receptors type 1, 3, and 5. And then the second type, the GI proteins, can be activated by muscarinic receptors 2 and 4. And want to learn more about GQ and GI proteins, make sure to check out my past videos on those. Everything's linked down below in the description. And then you have your nicotinic receptors, where you have 2 different branches, neuromuscular junction receptors and neuronal. NMJ receptors is involved in muscle contraction, more so skeletal muscles. And then neuronal receptors are involved in neurons and ganglias, especially in the parasympathetic and sympathetic nervous systems. Both of these actually are involved in parasympathetic and sympathetic nervous systems, and we will get into those in future videos, so don't worry. So that basically concludes response. Now, these responses have to be terminated to prevent them from going out of control, and that's where this last step, termination, comes into play. Let's get into it. Okay, so now, in order to terminate acetylcholine, the main way that it gets terminated is by enzymatic degradation. Now again, I know I've said this again and again, but I've already made a video all about termination mechanisms of messenger molecules, so be sure to check out that one. I've talked about enzyme degradation before, and I think it'll make it a lot more clear. That's also linked down below, along with all the other videos. But basically, so enzymes can come and they can act upon your acetylcholine to inactivate it or degrade it. And the most common of these enzymes is what's called ACE, which stands for acetylcholine esterase. Acetylcholine esterase. And what this enzyme does is it splits acetylcholine, which is your messenger molecule. It splits it via hydrolysis. So it creates 2 different components, acetate plus choline. So this enzyme ACHE is found in the synapse. So remember, the synapse is the gap between neurons, or a neuron and muscle. So like as soon as ACH is released into the synapse, this enzyme is here, and it gets to work appropriately to make sure that acetylcholine's responses don't get out of hand. So it splits it into these 2 components, acetate and choline. And when it does this, so let's draw. So it splits this into acetate and choline. So now you have acetate and choline both in your synapse. And remember we said at the start that inside your neurons, you have an enzyme, right, called choline acetyltransferase, which combines acetyl-CoA from mitochondria plus choline, and that's how you make acetylcholine in the first place. Remember we said that? Well, remember, acetyl-CoA, we know that comes from your mitochondria, but where does choline come from? Well, actually, the choline that we've just produced here in your synapse, it can get reuptaken into your neurons via a transporter. So it's like a cycle. Let's redraw it out to make it nice and clear. So you have your neuron, and inside your neuron, you have an enzyme called choline acetyltransferase, CHAT. This is the one we talked about before in our production stage. If you want a reminder, be sure to go back to the timestamp. But anyway, your choline acetyltransferase combines acetyl-CoA from your mitochondria, acetyl-CoA, not AA, from mitochondria plus choline to produce acetylcholine. And then that gets, as we said, it gets packaged, and then it fuses and it gets released, and it can go and activate nicotinic and muscarinic receptors, do different responses, and then it gets terminated by your ACHE, which is also found in this synapse to prevent acetylcholine from getting out of control. And what it does is it splits your acetylcholine into acetates and choline. Notice again that your acetate here is different than the acetyl-CoA that you started with, but your choline is the same. So your choline can actually get reuptaken into your cell via a transport mechanism. So the choline goes through a cycle. It's used to make acetylcholine, then acetylcholine is broken apart, and then it can get reuptaken by the neuron to make more. It gets recycled. And by doing this, by having your acetylcholinesterase break down your acetylcholine, you are preventing acetylcholine's response. You're preventing it from being able to bind to muscarinic and nicotinic receptors too much, and you're preventing the response from going out of control. So it's an effective termination mechanism. And that essentially covers the last step of our cell signaling pathway. We have successfully terminated acetylcholine. So it's important to recognize just how important termination is as a step. And in fact, there's some toxins that can interfere with this termination process. So here are some examples. Novichok is a chemical weapon. Sarin gas, this was used in the 1995 Tokyo subway terrorist attacks. And organophosphate is a chemical found in some pesticides. And all of these things are inhibitors of ACHE, acetylcholinesterase. They inhibit it. And without acetylcholinesterase, you cannot turn acetylcholine into acetate and choline. You can't break down acetylcholine. So it stops this process. And because of that, you have excess acetylcholine, which can do excess response. And this can be very dangerous. Excess acetylcholine can go and excessively cause muscle contraction, and this can lead to epileptic fits. And also, because acetylcholine is so involved in your sympathetic and parasympathetic nervous systems, excess amounts of acetylcholine can throw these systems out of line. And it can lead to irregular levels of digestion and heart rate and breathing rate, all of which can be fatal. Termination is an essential step for survival. So that basically concludes the acetylcholine pathway. We've reviewed how acetylcholine gets produced, how it gets packaged, and then eventually released, what responses it produces, and how it gets terminated, and how choline can be recycled and the cycle can continue over and over again. I hope you guys found this video informative and easy to understand. If there's anything else you would like me to go over, explain again, anything you found a little confusing, please let me know down in the comments. I'm willing to take any feedback and suggestions you guys have. But other than that, thank you guys so much for watching. I really appreciate all the support. In the next few videos, we're going to be looking at we're going to be doing kind of the same format of looking at the general cell signaling framework, but for some other molecules such as maybe adrenaline and norepinephrine and others. So stay tuned for that. But until then, thank you guys so much for watching. I'll see y'all next time. Take care.